Okay, this mini lecture is about the cytoskeleton component of cells. We're talking about cytoskeleton found in eukaryotic cells, though there are similar structures in prokaryotic cells. They're made up of different molecules, so this is focused on eukaryotes. So your three outcomes is to describe the general structure, properties, and functions of the different cytoskeleton components, microtubules, intermediate filaments, and actin filaments. Know the general role, of motor proteins for transport and which type of cytoskeleton they use. Know the difference between kinesin, dynein, and myosin. So these are your motor proteins. So the cytoskeleton is found throughout a eukaryotic cell. And this is showing you, and I'm going to abbreviate intermediate filaments as IF, microtubules as MT, and actin filaments are also called microfilaments. So this diagram is just showing you that they're located all throughout the cell. This is one of my most favorite pictures. This is called immunofluorescence. So it is a type of light microscopy, but we're using molecules that are stained that interact with specific molecules in the cell. So for instance, the microtubules in this picture are in green. The microfilaments are in blue, and a specific type of intermediate filament is in red. So again, you can see that these cytoskeleton components are found throughout the cell, and cytoskeleton is just protein, filaments. So we're going to look at these long structures all made out of different types of protein. One type of cytoskeleton that we've already talked about are centrioles. And centrioles are kind of seen it's hard to see exactly, but here, this is a dividing cell. So all the green is microtubules. So centrioles are a type of microtubule. And these long green pieces are called spindle fibers, which we will talk more about when we talk about mitosis and meiosis. These are also specific types of microtubules. And the function of a centriole is to help separate chromosomes, your DNA, during cell division. So in pink here, those stained molecules are actually your DNA chromosomes. And so what it's showing you in this image is that these microtubules are grabbing on to the DNA chromosomes, and then they're eventually going to move, to opposite directions in the cell and separate those chromosomes and eventually make two cells. So that's mitosis, and we'll talk about that towards the end of the semester. So we've already talked about centrioles. Now I want you to add to it that they are a type of microtubule, so they belong to the big class of structures as cytoskeleton. There are three main classes that make up cytoskeleton. So we said the microfilaments, and these are also called actin filaments because that is the protein that makes up the microfilaments. We have intermediate filaments. These are really specific to different cell types. They're very stable. whereas microfilaments and microtubules, we call them dynamic. Which means that they can grow and shrink all the time. And you'll learn lots about this in cell biology. Microtubules, again MT, they're dynamic. And one thing I want you to see is their names, well, kind of, maybe just intermediate. The microtubules are the thickest. So these are these hollow tubes, very thick, 25 nanometers. On the other end, microfilaments are very thin, 7 nanometers, and they're made up of actin, 2 strands. You can kind of see twisted here. And intermediate filaments are named intermediate because their size is in between the big microtubules, the small microfilaments, and they are also multiple protein filaments twisted together. So there's a chart to fill out to help you keep everything organized. So I would describe the structure of microtubules, which is down here, this first one, as a hollow tube. And it's made up of alpha and beta tubulin. That's the name of the protein type. And like I said, it's the thickest. Okay. So the function of microtubules is to maintain the shape of the cell, sorry, just look at my movement of chromosomes, like we talked about. So a specific example of that are the centrioles and the spindle fibers. They also help organelles move, which we call intracellular trafficking. And this is kind of like railroad tracks in a cell. So they're not, I mean, they're just called microtubules, but it's for movement of organelles. And vesicles. And another example of a microtubule, flagella and cilia are made of microtubules. And you know that those are for cell movement. Those are found on the outside of the cell, but they're the same tubulin proteins. Okay, change color here. Intermediate filaments, that's the one in the middle here. Their structure is made of 8 twisted protein fibers. And again, in cell biology, you'd get into more details about this. They have medium thickness. Their function is also structure, support, shape of the cell. And I just want you to know that they're very cell type specific. That's not really a function. Maybe we'll put that in an example. So they're cell type specific. And why I think this is interesting is if you have cancer that's metastasized, meaning cancer that's moved from its original location to somewhere else in your body, they can figure out where that cancer originated based on the type of intermediate filaments, because they're specific to specific cell types. So you could have a lump, a tumor in one of your lymph nodes that came from the liver, or came from the breast, or came from other types of tissue, and they can identify the origin of that cancer based on intermediate filaments. Keratin, that makes up your hair and nails, or if you were a bird, your beak. That's a type of intermediate filament. And you know your nails and hairs are pretty strong. Also, the nuclear lamina is basically the intermediate filaments that give the nucleus shape, right? So the nucleus is surrounded by a double membrane, and membranes are gushy and can move around. So to keep the shape and keep that DNA contained inside the nuclear envelope is this layer of intermediate filaments that give the nucleus its shape. Okay, last one is microfilaments. Down here, you can see that it's two intertwined chains. And it's made up of the protein actin. It is the thinnest. Very dynamic growth and shrinking. It's also for cell shape and organelle movement. But it's also for cell movement. So some cells can actually crawl, and they crawl by the microfilaments, the actin filaments, growing on one end and shrinking on the other end, and allows the cell to crawl forward. They're also important in cell division. So when you're dividing into two, they help push the plasma membranes away from each other and help the cell grow and separate into two different cells. Microfilaments are also important in muscle contraction. So when you learn some anatomy and physiology, and again cell biology, you'll learn about actin microfilaments and how they're important in muscle contraction. The other thing that goes along with the cytoskeleton is motor proteins. And motor proteins are proteins that move things in the cell. So the first one we have here is myosin, and it moves along actin filaments. So this is part of muscle contraction. And so now you have linked a motor protein, myosin, with cytoskeleton actin filaments. Over here, we have two different motor proteins. We have kinesin and we have dynin. And they both move along microtubules. Okay. So we've got microtubules associated with kinesin and dynein, actin or microfilaments associated with myosin. The big difference between kinesin and dynein is the direction of their movement. So dynein moves what we call retrograde, which if you're looking at a cell, it means towards the nucleus. Kinesin, on the other hand, moves anterograde, and that means it moves toward the cell membrane or the plasma membrane. So they're moving in opposite directions. Right? So if you have a cell, you can have kinesin moving things toward the plasma membrane with a K, and you can have dynein moving things towards the nucleus. And the things that they're moving are they're transporting. So, for instance, vesicles. And later in the semester, when we talk about protein synthesis, we're going to talk about vesicles and the endomembrane system and proteins moving from the ER to the Golgi. They move in vesicles with these motor proteins. Kinesin and dynein can also move organelles around to different spaces. So that's why I like to think of the microtubules as kind of the train tracks, and the dynein and kinesin are the trains moving their cargo. We're going to see if this works. This first video shows kinesin taking a vesicle. And remember, kinesin's going to move towards the plasma membrane. So it has a vesicle with all these little proteins, and it's chugging along this microtubule toward the plasma membrane. This video is showing a little bit about muscle contraction.
It just feels good next to my body, and I like the look that it gives me.
Myosin cross-linking occurs by a myosin head comprised of multiple proteins capable of conformational and positional changes. The myosin heads bind actin binding sites and undergo a conformational rearrangement in their proteins.
So myosin is the motor protein, and it's moving the cytoskeleton actin.
The conformational rearrangement rotates the position of the head and pulls the actin along the myosin. In the next scene, we will look at the biochemical events related to actin-myosin binding.
No, we're not. But what I want you to notice is both of them are using movement, and I mean both. All three of them are using movement, and all three of them, even though it doesn't show on this diagram, require energy, require ATP. So we'll talk about ATP being the energy of the cell. So just understand this takes energy, and it allows your cells and muscles to move. Okay, that's what I have for you for the cytoskeleton.